Hello, I'll be talking about grasshopper body size shifts in response to climate change. And this is part of a broad grasshopper resurvey project that is investigating genomic and physiological mechanisms of ecological and evolutionary responses to climate change. So we have a wide array of collaborators, and the resurvey project was led by Cesar Nufio. So declining body size has been termed a potential third universal response to warming. But most of the evidence comes from endotherms, and it comes from specimens that are rather haphazardly collected, often from the specimens that are found in museums. So to get straight to our punchline, we find that body size shifts are not a universal response. We find body size shifts that vary across elevations and life histories. So why might we expect variable body size responses for ectotherms? Ectotherms' body temperatures are highly sensitive to environmental temperatures, and there's also a strong temperature dependence of development and growth rates. So the majority of ectotherms follow what is known as the temperature size rule. So increases in environmental temperatures lead to increases in body temperatures. The biochemistry results in increases in both growth and development rates, but development rate increases are usually more of a pronounced response than increases in growth rate. So this results in decreases in development time and decreases in adult body size. However, quite a few organisms, particularly high elevation or high latitude populations of insects, often reverse this temperature size rule, and that is thought to allow organisms to capitalize on permissive conditions by growing bigger when conditions are warm. So we will be examining today a data set that includes femur length, which is a well-established index of grasshopper body size. It's less sensitive to short-term fluctuations, and we will be looking at data from more than 2,500 specimens that were collected at sites along an elevation gradient in Colorado that goes from Boulder up to Niwot Ridge and the Mountain Research Station. So we will be looking at specimens collected over 24 years, spanning the last century, and we will be comparing specimens from before and after 1980, with 1980 being a period at which climate change accelerated. So these specimens were largely initially conducted by Gordon Alexander and colleagues in the late 1950s, early 1960s. In the 2000s, Cesar Nufio came along and found these specimens sitting in dusty boxes at the corner of the museum. He basically rescued these specimens, curated them all, and to his, enthusiasm, he found weekly surveys that were recorded in notebooks. So these are sweep net surveys. A cool thing about grasshoppers is you can tell their developmental stage in the field quite readily. So he completed the survey or repeated these surveys along the elevation gradient over about 10 years. So we have a large degree of comparability across this time period. So let's start by examining the temperature size rule. So here we are looking at femur length as a function of elevation. We have data on the full grasshopper community, but we will be focusing on 6 species that are well represented along the gradient. So they are arranged here from early season species in the upper left to late season species in the lower right. And you will see that across all of the species, the slopes vary some, but you do see this relationship of organisms being smaller at cooler, higher elevation sites. So this is reversal of the temperature size rule that we might expect for a season-limited high elevation population, such as the ones that we are working on. You will also notice that females are substantially bigger than males, but they follow similar elevational lines. So we can then turn to looking at temperatures. We're fortunate because there is long-standing weather station data from our same sites. So we are looking at the mean of daily spring temperatures on the left and daily summer temperatures on the right across years. And we do find a lot of interannual variability, but we do also find significant warming that is focused at high elevation sites. So we can then ask, how do grasshopper body sizes vary over recent decades and in response to seasonal temperature variation? So we are trying to look at potentially evolutionary responses as well as shorter term developmental plasticity responses to temperature variation. And we want to know how these responses vary among species with different seasonal timing and across elevation. So based on our previous field and lab work, we can come up with conflicting hypotheses for what we expect. But overall, we concluded that based on the temperature size role reversal of grasshoppers being larger, warmer, lower elevation sites, we expect that warming would increase body size. And we do see greater developmental plasticity, particularly of the early season species at higher elevations. So we expect that size responses will be more pronounced at high elevations. So what did we find? Here we are looking at femur length anomaly. So basically we want to take out that elevation trend. So we are focusing on differences from mean body sizes across species, site, and elevation. Looking at the same 6 species arranged from early to late species, we do find a significant main effect of body size getting bigger over time as we expected, but we also find significant interactions with elevation and with species. So somewhat contrary to our expectations, we find that size shifts are focused at low elevation. If you look at the early season species on the left here, you can see that they are getting bigger. If we look at the later season species on the right, they are getting smaller. So next, we wanted to look at the role of seasonal temperatures and potentially accounting for these size differences. So here we are looking at femur length anomaly as a function of temperatures in the spring and temperatures in the previous summer. And we do find a significant role of seasonal temperatures, again, potentially indicative of developmental plasticity, but we do not fully account for the time effect using temperatures. So that suggests the potential for a longer term, potentially evolutionary responses, but there could be a lot of other factors accounting for our inability to account for the full time difference, such as inaccuracies and how we characterize the environment. But our models suggest that warmer spring temperatures increase size. This is consistent with developmental responses. For nymphal diaposers, we find that warmer temperatures the previous summer decrease size, and this is potentially consistent with increased rates of energy use in the warmer conditions. Interestingly, we find that thermal responses are most pronounced at high elevation, so that's consistent with our expectation based on developmental plasticity, but it does suggest that perhaps the plasticity is acting to maintain similar body sizes at high elevation rather than resulting in body size shifts. So finally, we wanted to look at the link with seasonal timing. We have 2 different metrics of seasonal timing that show quite similar trends. So the first is estimates of phenology from our field data, but what we're showing here is the date that specimens were collected. So how does body size vary as a function of day of adulthood in anomaly in terms of when specimens were collected? We again find quite a bit of scatter, but we do find quite a few species showing significant diagonal relationships here. So that is consistent with grasshoppers, particularly nymphal diaposing or early season species tending to capitalize on warm conditions by both getting bigger and having earlier phenology. So again, our overall message is that insect sizes have not responded universally to climate change. Shifts vary across elevations and life histories. And this is another reminder of the need to consider physiology and how it varies across elevations and life histories when projecting organismal responses to climate change. So thanks to NSF for funding the research. Thanks to the Museum of Natural History for curating, providing access to the specimens. Thanks to the many CU undergrads who helped measure specimens. Thanks to my collaborators, and thanks to you for your interest.